Hi, my name is Zakari, and today I'll give you a brief overview of the biosynthesis of cholesterol as well as the degradation. Cholesterol is a biomolecule important for many different bodily functions. However, excessive amounts lead to many different diseases such as cardiovascular disease and hypercholesteremia, thus the need to regulate the process. This is done by a transcription factor, SRB protein 2, which controls the rate of enzyme expressions important in the biosynthesis process. Starting with the process, we have acetyl-CoA molecules. Three of these molecules are combined into HMG-CoA by the activity of HMG-CoA synthase. HMG-CoA is then converted into mevalonate by HMG-CoA reductase. Mevalonate needs to be activated, and it is done so by adding ATP molecules to create an isopentenyl biphosphate. Five of these molecules are then converted into squalene. Squalene is then combined into lanosterol, and lanosterol is converted into cholesterol. Now, this cholesterol needs to be degraded because, like I said earlier, excessive amounts lead to different diseases. So the body regulates this process by transporting cholesterol to the liver where it can be converted into bile salts. However, excessive bile salts is acidic for the body, so it converts these bile salts into feces.
Now that we've discussed cholesterol synthesis, let's talk about cholesterol transport. Cholesterol is transported by two distinct pathways. The exogenous pathway is dietary cholesterol, and dietary cholesterol is cholesterol-rich foods that is ingested and lands in the small intestines, where it forms chylomicrons. Chylomicrons are lipoproteins that transport cholesterol out of the intestines into the blood and then eventually to the liver. Now we've reached the second pathway, the endogenous pathway. Cholesterol that's synthesized in the liver is exported by another form of lipoproteins called very low density lipoproteins. Very low density lipoproteins sends cholesterol to adipose tissues and muscle cells. After cholesterol is sent to these tissues and cells, it converts to low-density lipoproteins that sends the remainder of cholesterol to peripheral cells. Excess cholesterol transports back to the liver by high-density lipoproteins.
Once cholesterol is transported throughout the body, there are 5 distinctive cellular processes and properties that it aids in the human body. For instance, like Tamara just mentioned, cholesterol is returned back to the liver, and here is where it is converted to bile salts. And bile salts are used to emulsify fatty acids. The second function of cholesterol is it provides fluidity of the cell membrane. So here at high temperatures, the lipid bilayer is more fluid and cholesterol is more rigid. However, at low temperatures, the lipid bilayer is more rigid and cholesterol is more fluid. A third function of cholesterol, it is the precursor of all our steroid hormones. So here we have a diagram showing the synthesis of our hormones starting from the precursor, which is cholesterol. The fourth function of cholesterol is that it acts as an insulator in axons. So here our myelin sheets are rich in cholesterol, and they act as an insulator and an inducer of the electrical signals between the cells. Last but not least, a fifth function of cholesterol is that it leads to the synthesis of vitamin D.
Hi, my name is Michael Tucker, and today I'm going to talk to you about 2 drugs that are used in the inhibition of cholesterol synthesis. So first and foremost, in the synthesis of cholesterol, 2 acetyl-CoA molecules are required to form an acetyl-acyl-CoA molecule. Next, with the help of HMG-CoA synthase enzyme, and also a third acetyl-CoA molecule, those create HMG-CoA. Now, to create mevalonate from HMG-CoA, HMG-CoA reductase enzyme is required. Now, HMG-CoA reductase, that's the key rate-limiting enzyme in cholesterol synthesis, and so it's a major regulatory enzyme, and so statins, they will inhibit this enzyme to effectively lower the total cholesterol within the body. The second type of drug I'm going to speak to you today about are squalene synthase inhibitors. Now, squalene synthase inhibitors are exactly what they say they are. They inhibit squalene synthase. And what squalene synthase does, it dimerizes two farnesyl pyrophosphate molecules to afford a squalene. And now squalene is one of the molecules that you have to to synthesize cholesterol. And so if squalene synthase enzyme is inhibited, then there will be no cholesterol synthesis at all.